Hello everyone, good evening. Welcome to today's webinar. And today's webinar is all about the physicochemical properties of the drug molecules, which comes under your Physical Pharmacy 1. And I'm Mrs. P. Nagharita, Associate Professor from the Department of Pharmaceutics. And we have gone with few webinars earlier. And today we are going to see about the physicochemical properties of the drug molecules. For any drug molecule that is present, how its physicochemical properties. The physicochemical properties are nothing but the combination of both physical as well as the chemical properties. Like for example, if you consider the physical properties, it is like melting point, boiling point, all these comes under the physical properties. And chemical properties are like the structure and how it is going to affect the drug activity like that. So the combination of both these physical and chemical properties comes under the physicochemical properties. And how this physicochemical and why these physicochemical properties are important in order to understand about the drug's activity, right? So that we are going to see in today's webinar. So coming to a physicochemical property, the definition here it goes. So the ability of a chemical compound to elicit a pharmacological or therapeutic effect is related to influence of various physical and chemical properties of the chemical substance on the biomolecule. Okay, the on whatever the drug molecule that you have taken, this particular drug molecule's pharmacological activity or the therapeutic response, it is going to depend on the physical and chemical properties or the physicochemical property of the drug molecule. So the physical properties of a drug is responsible for its action, whereas chemical properties are extracellularly according to the simple chemical reactions like neutralization, chelation, and oxidation. So how a particular drug is chemically reacting and by altering its physical property, how it is going to show the pharmacological action. Right? So these are the various physicochemical properties that are available starting from solubility, partition coefficient, dissociation constant, hydrogen bonding, ionization of drug, redox potential, complexation, surface activity, protein binding, and isostericism. Almost we have some 7 to 8 types of the physicochemical properties that we are going to study today. So first, let us start with the solubility. So what is solubility? Solubility is nothing but the ability of a solute molecule to get dissolved in the solvent without any particular external energy. It is called as a solubility. So at any given temperature, it is defined as the concentration of dissolved solute, which is in equilibrium with the solid solute or the solvent. So it depends upon the nature of the solute and solvent as well as temperature, pH, as well as the pressure. So how much amount of the solute is getting solubilized in the solvent? It depends upon the temperature, pH, as well as the pressure. Because if they are going to change in any of these properties, either temperature, pH, or the pressure, definitely the solubility of any drug is going to be changed in any particular solvent. The solubility of drug may be expressed in terms of its affinity or philicity. Affinity or philicity is nothing but attraction towards certain molecules is called as a affinity. Or repulsion, we also call it as a phobicity, like hydrophilicity, hydrophobicity, or lipophilicity, like that we call it as. Philicity is nothing but affinity and phobicity is nothing but the repulsion for either an aqueous or organic solvent. So like if your drug is soluble in the water, we call them as a hydrophilic. If they are not soluble in water and if they are not properly soluble in water, then we call it as a hydrophobic, like that. So in that way, we express the solubility terms. The atoms and molecules of all organic substances are held together by various types of the bonds like hydrogen bond, dipole-dipole bond, ionic bond, etc. And these forces are involved in solubility because it is the solvent-solvent, solute-solute, solvent-solute interactions that governs the solubility. That means here, when the drugs are held together by certain bonds called as a hydrogen bond or dipole-dipole or ionic bonds, here, the force required or the energy required to break these bonds so that the solute get solubilized with that of the solvent, it depends upon these interactions, solute-solute interactions, solvent-solute interactions, as well as the solvent-solvent interactions. So when there is a high solute-solvent interaction, then you require more energy to break that solute-solvent interaction. Okay, so in that way, the solubility depends upon these type of the bonds. And there are certain methods which are used to improve the solubility, because there will be certain drugs which do not have a proper solubility. And the solubility of such type of the drugs can be enhanced by various methods. Here I have given only few methods, because that is not the point here. This you'll be studying in detail in the future classes. Here, the structural modification, that is, you're going to alter the structure of the molecule such that you're going to enhance the solubility of any drug in the solvent. And use of co-solvents. Co-solvents are nothing but these are another type of the additive substances which are added to the solvent so that it will help in the solubilization of the solute. And then using the surfactants. Surfactants, these are the amphiphilic substances which have both polar as well as a non-polar structure, and it will help in solubilization of the solute in the solvent. And then complexation. Complexation here is nothing but when a substance is not able to solubilize within the solvent by its own, you're using another agent called as a complexing agent so that it forms a complex. And now this complex can be easily solubilized in the solvent that you have taken. That is called as a complexation. And still we have many more methods for improving the solubility of the drugs. But just as an example, here I've given you the four types. So why actually we have to study about the solubility and how the solubility is important in the in eliciting the pharmacological action of any drug. So the solubility concept is important to pharmacists because it governs the preparation of liquid dosage form, and the drug must be in solution before it is absorbed by the body to produce a biological activity. That means in two conditions, this solubility is very important. The first one is while preparing the liquid preparations, like whenever you are preparing the liquid preparations, at what concentration the solid is going to be dissolved in the solvent you are going to see, and also when you have prepared the solid dosage form, at what temperature or at what pH the drug is undergoing dissolution, because you know that inside the body, the drug will be absorbed only in the solution form. It is not going to get absorbed in the solid form. So you should know at what pH or at what temperature the drug is undergoing the solubility. That means conversion of the drug from solid into the solution form. And drug must be in solution form to interact with the receptors as well. So that is all about the solubility, why the solubility is required to be studied in order to know its effect on the pharmacological action of any drug. The second one is the partition coefficient. So the partition coefficient is nothing but the ability of the drug to distribute among the aqueous layer and the lipid layer, right? So here, why we should know about this partition coefficient is nothing but whenever the drug has been administered into the body, you know that all the membranes inside the body are lipophilic in nature. Okay, solubility is there for showing or for determining the dissolving of the drug will be solubilized inside the body and it will be releasing the drug. Now, the second step for the drug to get absorbed is the permeation. Permeation is nothing but the ability of the drug to cross the membrane is called as a permeation. So that permeation is always dependent upon the partition coefficient. That means how much amount of the drug is being partitioned between the lipid molecules or between the lipid layer and the aqueous layer. Aqueous layer is nothing but your GI contents, and the lipid layer is nothing but the membrane through which it has to cross the membrane. Okay. So here drug aqueous and drug lipid. So how much amount of the drug is being distributed between the aqueous layer and the lipid layer, it is governed by the partition coefficient. So it is one of the physicochemical parameter which influences the drug transport and drug distribution, the way in which the drug reaches the site of action from the site of application. Application, I have taken the drug. Site of action, that means maybe at liver or lungs, it has to go. So it has to cross certain membranes. So how much amount of the drug that I have applied at the site of application and how much of the drug is reaching the site of action. Partition coefficient is defined as the equilibrium constant of drug concentration for an unionized molecule in two phases. So what are those two phases? Lipid and water are the two phases. And this for ionized molecule here, the partition coefficient can be determined by drug in lipid layer divided by 1 minus A concentration of drug in water, where A is the degree of ionization in aqueous solution. And it affects the drug transfer characteristics, obviously. When the drug is having a good partition coefficient, it can easily cross the lipoidal membrane and it can reach the target site. However, if it is not having the proper partition coefficient, then it cannot cross the membrane properly. The contribution of each functional group and structural arrangement help to determine the lipophilic or hydrophilic character of the drug molecule. So this partition coefficient depends upon all the functional groups and the structure of the drug molecule, which is present in the structure of the drug. And that particular structure of the drug determines whether it is a hydrophilic drug or a lipophilic drug. If it is having a higher partition coefficient, it is called as a lipophilic drug. If it is having a lesser uh, partition coefficient, then it is called as a hydrophilic drug. And this type of the classification is mainly used in QSAR. That is a quantitative structure activity relationship. So there are certain factors that are going to affect the partition coefficient. They are the pH, co-solvent, surfactant, and the complex section. So how they are going to affect the pH? So for example, if the drug is affected by any pH. So by altering the pH of that drug microenvironment, you can enhance the permeability of the drug and the co-solvents that have been used as well as. So here if you see the co-solvents, surfactants and the complexation, all these are the methods which we used in enhancing the solubility. So when the solubility has been always the solubility and permeability are like rivals. If solubility is enhanced, that means the hydrophilicity has been attained. So when hydrophilicity has been attained, the partition coefficient would become low. That means the ability for a drug to cross the membrane would become low. So it becomes a hydrophilic structure. And again, you have to take certain measures to improve the lipophilicity. For that reason, any drug should have a proper HLB, hydrophilic lipophilic balance should be there. It should not be neither more nor less. So in general, this partition coefficient is difficult to measure within the body. That is in vivo, it is difficult. Whereas in vitro, it is generally measured by using the octanol and water system. Okay, why only octanol is used here? Because it has both polar and non-polar region and it is easy to measure and it often correlates with many biological properties and it can be predicted using the computational mode. So that is the reason we take octanol as a standard in order to determine the partition coefficient of any drug. The partition coefficient P is dimensionless, whereas its logarithm log P is widely used for the measure of their lipophilicity. And there are two methods which are used to determine this log P value. They are the shake flask method. This is the most common method and you must have done in your practicals as well. And then the chromatographic method, especially by using the HPLC method. So phenobarbital has a high lipid water partition coefficient of 5.9 and thiopentone sodium has a chloroform water partition coefficient of about 100. So it is highly soluble in the lipid. So here thiopentanone sodium is used as a ultra short acting barbiturates. So what happens and what are the various factors that are affected by the log P? If there is increase in the log P, there will be increase in the binding to the receptors. increase in the binding to P450 enzyme, increase of absorbance through the membrane, and increase of the binding of drug to the blood or tissue proteins. And only there is one parameter that is going to decrease with increase in the log P is the aqueous solubility. So more aqueous solubility, less log P, more the log P, less the aqueous solubility. As I said, it is completely like rivals. So when there is increase in the solubility, there will be decrease in the log P value. If there is increase in the log P value, there will be decrease in the solubility. So other than the solubility, other all parameters will be increasing with the increase in the log P value. So why this partition coefficient is important? So it is generally used in combination with the pKa, that is ionization constant, to predict whether the distribution of the drug in the biological system is proper or not. Because as a biological system consists of various types of the membranes in the body, this distribution always depends upon the log P value, that is the tendency of a drug molecule to cross these membranes. The factors such as absorption, excretion, and penetration of the CNS may be related to the log P value of the drug, because all those are the lipophilic membranes. The drug should be designed with the lowest possible log P to reduce toxicity, non-specific binding, as well as the bioavailability. So because why we go with the optimum log P value, because as the log P value increases, the solubility is going to decrease. That is why we should always have the balance between the hydrophilicity and the lipophilicity. So this is all about the partition coefficient. And then the next one we have is the hydrogen bond. So it is a special dipole-dipole interaction between the hydrogen atom in a polar bond, such as NH, OH, FH, and electronegative atoms like oxygen, nitrogen, and fluorine atoms. So these dipoles result from unequal sharing of electrons between the atoms within the covalent bond. And these hydrogen bonds are the weakest bonds, which are denoted by the dots. It is not even denoted by a straight line, it is denoted by the dots. And the compounds that are capable of forming hydrogen bonding is only soluble in the water. If they are having the hydrogen bonds, then you can blindly say that they are soluble in the water. And there are two types of the hydrogen bonding: intermolecular hydrogen bonding and intramolecular hydrogen bonding. So intermolecular hydrogen bonding is nothing but the formation of the bonds between two like chemicals, two like drugs, is called as a intermolecular. Intramolecular is nothing but within the same compound, the formation of a hydrogen bond, then it is called as a intramolecular hydrogen bond. So here it is the intermolecular hydrogen bonding. It occurs between two or more than two molecules of the same or the different compounds. And due to the increase in the boiling point of the compound and increase the molecular weight of the compound, so more energy is required to dissociate the molecule from the vaporization. And then intramolecular hydrogen bonding occurs within two atoms of the same molecule. This type of bonding is also called as a chelation, and it frequently occurs in the organic compounds. And sometimes this hydrogen bond develops 6 or 5 member rings because of the formation of the hydrogen bond. And this is formed because of the reduction in the bonding. So here it is the salicylic acid and it is orthonitrophenol. So within this, if you see, there is a formation of 1, 2, 3, 4, 5, 6. 6-membered ring structure has been formed. The same has been followed here. Okay. So for this reason, it is called as it is forming within the molecule, within the atoms of the same molecule, it is called as an intramolecular hydrogen bond. So here hydrogen bonding, how it is going to affect the biological action is nothing but depending upon the type of the bond that has been formed, the biological action has been changing. So here antipyrin, if you see here, it has antipyrin is generally 1-phenyl 2,3-dimethyl 5-pyrazolone. And when the this particular hydrogen bond has been formed, it is forming it as 1-phenyl 3-methyl 5-pyrazolone. So the first compound, it is having analgesic activity, whereas the compound which is formed here, it is inactive. It is not having any analgesic activity. And then another example we have here is the salicylic acid. And here again, when this salicylic acid is forming the hydrogen bond, we are not having any Initially, it is having the antibacterial activity, but when it is forming the hydrogen bond, it is becoming inactive by this para and meta hydroxy benzoic acids. And then what are the properties that are going to be affected because of this hydrogen bonding is boiling point and melting point, water solubility is affected, strength of the acid, spectroscopic properties on surface tension and viscosity. It is going to show the effect biological products as well as the drug receptor interaction. All these properties are going to be affected if there is a formation of a hydrogen bond. And then the next property that we have here is a chelation or complexation. So the chelation or complexation is nothing but the tendency of any drug molecule to form a complex with the agent called as a complexing agent. It is called as a complexation, or also it is called as a chelation. So the complex of drug molecules cannot cross the natural membrane barriers that render the drug biological ineffectivity. So when this complex has been formed, the drug cannot be it cannot cross the biological membrane. And when it is not able to cross the biological membrane, it is not able to show the effect. Okay, it is not able to show the biological effect. So the rate of absorption is proportional to the concentration of free drug molecules. That is the diffusion of the drug. So always the amount of the instead of calling the intensity of the drug molecule to show the biological action depends upon how much amount of the or how much concentration of the free drug molecules are available to show the action. If free drug molecules are not available to show the action, if the complexes has been formed, then your biological activity of that particular drug will also be reduced. So due to reversibility of the complexation, equilibrium between the free drug and the drug complex. So this is a reversible reaction. It is not an irreversible reaction. So the drug plus complexing agent, this is a reversible reaction where it is forming a drug complex. And the complexation reduces the rate of absorption of drug but not affect the availability of the drug. So the amount of the drug that is responsible for getting absorption is going to be affected, but the bioavailability of the drug is not going to be changed because of the complexation property. So here, importance of chelates in medicine is it is used as an antidote for metal poisoning. So whenever a metal poisoning is happened in the body, if an antidote is injected inside the body, so that poison will form a chelate or the complexing agent with this antidote, and it will restrict moving into the systemic circulation. So when your poison is not able to enter into the systemic circulation, it cannot affect the system inside the body. Okay, so here dimercaprol is used as a chelating agent. This is the arsenic, which is a metal poison. This dimercaprol, it is going to be forming as a complex with the arsenic, and it is forming the complexing agent. This particular complexing agent, it cannot move the biological membrane, and it is going to stop the toxic properties of the arsenic. Okay, and the next one is a penicillamine is another such example of the chelate. 8-hydroxyquinoline and its analogs acts as an antibacterial and antifungal agent by complexing with the iron and the copper. These are another examples again. And undesirable side effects caused by the drugs, which chelates with the metals are a side effect of hydrolysine, a antihypertensive agent, is formation of anemia, and this is due to chelation of the drug with iron. So whenever this hydrolysine, which is called as the antihypertensive drug, is injected or it is administered to the body, the side effect called as the anemia. What is the anemia? Like lack of iron in the blood. So, when this hydrolysine has been taken as an antihypertensive drug, there will be reduction in the iron in the patient's body because this hydrolysine, it is going to form a complex with the iron that you have used inside, iron that is produced inside the body from the blood. Okay, so when it is binding method of the iron, now iron is not available in the blood. That is why there will be a condition called as a anemia. And phenobarbital forms a non-absorbable complex with polyethylene glycol 4000 and calcium with EDTA. This is your most favorite example when we consider the complexing agent. So calcium with EDTA, it forms a complex, and also it increases the permeability of the membrane. right? So this is about the chelation or the complexing agent. And then the next fifth. So just to go back about what are all the properties that we have studied till now. So the first one is the solubility we have seen, and the second one is the partition coefficient. Third one is the hydrogen bond, and the fourth one is a complexing agent or the chelating agent that we have seen. And then the next most important physicochemical property is the ionization of drug. So most of the drugs are either weak acids or base and can exist in either ionized or unionized state. So what is ionization? Ionization is nothing but protonation or deprotonation that results in the charged molecules. Okay, it results in the charged molecules. And the ionization of the drug, it depends upon its pKa and pH. So what is the pKa? It is the ionization constant, and pH is nothing but at what pH, whether it is a acidic pH or a basic pH. Okay. So the rate of drug absorption is directly proportional to the concentration of the drug at absorbable form, but not the concentration at the absorption site. So always you should remember, this is a very important point that you have to remember is the rate of drug absorption is directly proportional to the concentration of the drug at absorbable form. So whether it is in absorbable form or not, that you are going to see, whether it is in a ionized form or unionized form. It is not required that how much amount of the drug is present in the site of action. The absorbable drug, that is, the drug is available in the form in which it can be absorbed. So ionization form imparts good water solubility to the drug, which is required of binding of drug and receptor interaction. And also unionized form helps the drug to cross the cell membrane. Again, controversy has come. If it is in the form of ionization, it gets good solubility. And if it is in the form of unionized drug, it can cross the membrane easily. So example, barbituric acid is inactive because it is a strong acid. And while 5,5 substituted barbituric acid has CNS depressant action because it is a weak acid. So here again, you are going to see what happening when the drug is in the form of an ionized drug and when it is in the form of an unionized drug. So here I have just given the example of how unionized drug is becoming as a conjugate acid and a base, and ionized drug is becoming as a conjugate acid and a base. So here we take the equation called as a Henderson-Hasselbalch equation, a very important equation. So for acids, pH minus pK is log concentration of ionized by unionized, whereas for the base, it is pH minus pK is equal to log concentration of unionized by ionized. So percentage of ionization can be calculated by 100 divided by 1 plus 10 to the power pH minus pK. So when an acid or a base is 50% ionized, then pH will be equal to the pK value. So the solution of weak acid aspirin in the stomach will get readily absorbed because it is in the unionized form. 99% of the drug is present in the unionized form. So it can be easily absorbed from the stomach whose pH is in the acidic range itself. So always you should remember that if the drug is in the form of an ionized state, the solubility will be easier. And if the drug is in unionized state, the permeation will be easier for the drug. Phenytoin injection must be adjusted to pH 12 with sodium hydroxide to obtain 99.98% of the drug to be in the ionized form. And tropicamide eye drops, an anticholinergic drug, has a pK of 5.2, and the drug has to be buffered to pH 4 to obtain more than 90% of the ionization. So what is the importance of this ionization of the drug? Weak acid at acidic pH, more lipid soluble because it is uncharged, and the uncharged form more readily passes through the biological membrane. So that is what we have studied. And weak base at alkaline pH, more lipid soluble because it is uncharged, and the uncharged one will be passing through the biological membrane easier. So always when the drug is in the unionized form, it can be easily crossing the biological membrane. Okay, so that is about the pKa ionization constant. And then the next one is the redox potential. So the redox potential is nothing but the oxidation and the reduction reactions together. We call it as a redox. So the oxidation reduction potential may be defined as a quantitative expression of the tendency that a compound has to give or receive electrons. So what is the ability of a compound to either donate or receive the electrons is called as a redox potential. So the correlation between redox potential and biological activity can only be drawn from the compound of very similar structure and their properties. So it can be calculated from the equation E is equal to E naught plus 0.0592 divided by N log concentration of the reductant divided by concentration of the oxidant. So here riboflavin analog is an example. The biological activity of riboflavin is due to E is equal to minus 0.185 volts. The another one is a and the another one is a dichlororiboflavin whose E naught value has been increased drastically. And then the bacteriostatic activity in quinones, it is associated with the redox potential whose redox potential is at plus 0.03 volts when tested against the Staphylococcus aureus. So that is how the redox potential is going to identify at what about the biological activity of any drug depending upon the redox potential. And the next one is the surface activity. So it is defined as a material that can reduce the surface tension of water at very low concentration, just like a surfactant. Okay, so the surface active agents affect the drug absorption, which depends upon the chemical nature of the surfactant. Like we have various types of the surfactant, ionic surfactants, non-ionic surfactants. Okay, so like that, depending upon that, the chemical nature of the surfactant, you're going to see whether it is affecting the drug absorption or not. Its concentration, that means in how much concentration you are using that particular surfactant, and its effect on biological membrane and the micelle formation. So the micelle formation is nothing but the minimum amount of the surfactant that is required to form the micelle. You must have studied this under critical micellar concentration. So that is the minimum amount of the surfactant that is required to form a micelle is called as a critical micellar concentration. And in lower concentration of surfactant, enhance the rate of absorption because amphiphiles reduce the surface tension and it shows the better absorption. Obviously, this surfactant is the most simplest form and the most, mostly used form of the method which is used to reduce the surface tension between the water and the drug molecule. So when these surfactants are used, it get easily solubilized and then getting permeated through the membranes. Whereas when you use this surfactants in excess quantity, then it is going to absorption. It is going to reduce the absorption of any drug. So here the applications include anti-helminthic activity of hexyl resorcinol. That means it has been enhanced by using the surface active agents. Bactericidal activity of cationic quaternary ammonium compounds, bactericidal activity of aliphatic alcohols, disinfectant action of phenol and cresol. All these have been increased with the increase in the by using certain surfactants to increase the solubility of any drug. And then the next one, so the reversible binding of protein with non-specific and non-functional site on the body protein without showing any biological effect is called as a protein binding. It is nothing but when a drug comes and bind with that of the intrinsic proteins. Intrinsic proteins are nothing but the proteins that are available within the body. So when your drug is coming and interacting with that of the protein, then it is called as a protein drug complex. And depending on whether the drug is a weak or strong acid, base, or it is a neutral, it can bind to single blood proteins to multiple blood proteins. So what are the various blood proteins that we have? Serum albumin, alpha acid glycoprotein, lipoproteins. And in lipoproteins, again, we have low density lipoproteins and high density lipoproteins. And the most significant protein is the albumin because it is the most majorly available protein in the body and it comprises more than half of the blood proteins. That is, it is a majorly available protein that is available in the body. Protein binding values are normally given as the percentages of the total plasma concentration of drug that is bound to all the plasma protein. So, free drug plus free protein, drug protein complex. This is how you are going to depict the protein binding of the drug. Next one is the stereochemistry of the drugs. It involves a study of three-dimensional nature of the molecules, and it is a study of chiral molecules. We are not going in depth about the stereochemistry here. We are just going to see how the stereochemistry of the drug is going to affect the action of the drug. Okay, it plays a major role in the pharmacological properties because any change in stereospecificity of the drug will affect its pharmacological activity, obviously. If it is rotated, levo-rotated or dextro-rotated, because of the change in the stereospecificity of these drugs, there will be change in the pharmacological action. That is why we are very much particular about the stereochemistry of the drug. And the isomeric pairs have different physical properties like log P, pK, etc. That means the two different isomers have different physical properties as well as the chemical properties. That is why it is affecting the pharmacological activity of the drug. And the isomer which have same bond connectivity but different arrangement of groups or atoms in the space are called as a stereoisomer. So this is the definition of a stereoisomer. And what are the conformational isomers? The different arrangement of atoms that can be converted into one another by rotation about a single bond, they are called as the conformations. And rotation about bonds allows interconversion of the conformers. So here we have given just few conformers like staggered conformers, eclipsed, Gaussian, fully eclipsed, like that, various amount, various rotations of the acetylcholine which can exist in different conformations. And then optical isomers. Stereochemistry, enantiomers, symmetry, and chirality are important concepts in therapeutic and toxic effect of the drug because when there is a change in the stereochemistry of the drug, sometimes there may be a change in the pharmacological action, and if it is more than the required state, it is going to cause some toxic effects or adverse reactions for the. So a chiral compound containing one asymmetric center has two enantiomers, and although each enantiomer has identical chemical and physical properties, they may have different physiological activities like how they interact with the receptors, how they undergo metabolism, and even the protein binding. And optical isomer in biological action is due to one isomer being able to achieve a three-point attachment with the receptor molecule, while its enantiomer would be able to have only two-point attachment with that of the same molecule. So in this way, how the stereochemistry is going to affect the biological action of the drug. When it is in one form, it will be able to bind to the protein in only to some extent, whereas when it is converted to the another form, there is a total change in the binding of a drug to the protein. So, for example, adrenaline, where we have here levo and the dextro adrenaline slides. So, how it is being attached to the receptors. So, here if you see there is 4-point interaction, 1, 2, 3, 4. Whereas here if you see there is only 3-point. Or else if you consider this complete thing as a 1-point, there is only 3-point and 2-point interaction with the receptors. This is how the change in the isomers, it will be affecting the biological action. So another example is the ephedrine and pseudoephedrine. So the category of drugs where the two isomers have quantitatively similar pharmacological activity but have different quantitative potencies, it comes with the warfarin. And then geometric isomerism, here also trans-diethyl sylvestrol and cis-diethyl sylvestrol. Here it is having an estrogenic activity, but whereas 7% activity will be seen with the trans isomer. And then the next one we have is the isosterism. Longmore introduced this term isosterism in 1990, which postulated that two molecules or molecular fragments containing an identical number and arrangement of electrons should have similar properties, and they are called as a isosters. And these isosters should be isoelectric. That means they should contain same number or same total charge. Okay, so this bioisosterism is defined as compounds or groups that possess near or equal number, shapes, and volumes, approximately the same distribution of electrons, and which exhibit similar. That means just like the identical twins. Okay, so how they have everything similar. And they are classified as a classical bioisters and non-classical bioisosters. And the classical bioisosters, these have similarities of shape and electronic configuration of atoms, groups, and molecules, which they replace. And the classical bioisosters, they may be univalent or the bivalent. So these are the examples that we have, the different types of the functional groups which are considered as a bioisosters, and trivalent, tetravalent ring structures we have. And then how this application of classical bioisosters, it is in the so replacement of NH2 group by the CH3 group. So when NH2 is there, we call it as a carbutamide, and CH3 is there, we call it as a tolutamide. Okay, so like that, OH with SH. When OH is there, it is a guanine. When SH is there, it is a 6-thioguanine, right? And then non-classical bioisosters, they do not. Non-classical bioisosters have valency electron configuration, and these have specific characteristics like electronic properties, physicochemical properties of the molecules, special arrangement of the molecules, and even the functional moiety for the biological activity. And these are the examples of various non-classical bioisosters like halogens, ethers, carbonyl groups, hydroxyl groups, catechols, right? And trans diethyl silvestrol and 17 beta estradiol, it comes as a non-classical type of the bioisosters, right? So, excuse. These are the various types of the physicochemical properties of the drug molecules and how they are going to affect the biological action of any drug. This is what we have, and almost 10 physicochemical properties we have seen. This isosterism, as such, though it is not actually studied under the physicochemical, but this also have certain effect on the biological action of the drug. So I have included that in the basic principles. So now, if anyone is having any doubt in today's class, in today's webinar, you can please post the questions in the chat box. If you have any doubts, please post your questions in the chat box, and please go through thoroughly about this PPT, and you can like easily answer the quiz questions that will be posted on this particular topic. If you have any doubts on this, so I think we have covered all these topics: solubility, partition, coefficient, dissociation, hydrogen bonding, ionization, everything is done. Okay, so if you have any doubts in today's topic, you can please post your questions in the chat box. I'll be happy to answer those questions. Yes, may I please know if anyone have any questions on this topic? All right, expecting that there are no questions on this topic, let us stop today's webinar. Thank you so much, everyone, for attending today's webinar. See you soon.